Your body takes energy from the fuels you eat and converts it to ATP. But in order to get there, it often has to go through NAD. Problem is, NADH can't actually get into the mitochondria. It's into the inner part of the mitochondria, the mitochondrial matrix. So mitochondria are the little compartments inside of your cells, sometimes referred to as the powerhouses of the cells, where the big energy payoff comes into play, where you can convert the electrons, take the electrons from the NADH and actually use them to power the making of ATP through the electron transport chain. And so a lot of the processes that ultimately generate ATP, along the way, what they're actually generating is NADH. They're basically taking electrons from different food sources and shuttling them through NADH to the electron transport chain, which is the process taking place in the mitochondria, or the powerhouses of your cell, where the bulk of the energy making is going to happen, where you're taking those electrons from those different energy sources and using the passing of those electrons between different molecules from one thing that doesn't want it that much to something that wants it more, to something that wants it more, to something that wants it more. And each time you're passing it to something that wants it more, you're making the molecules more happy. When you make molecules more happy, you're releasing energy. And in this case, you're using that energy to drive the protons through a membrane, to cause a buildup in the concentration of protons in this like inner membrane space. And then those protons are going to flow through this little pump, this ATP synthase that's going to turn this little motor and make ATP. So it's really, really awesome. And it's relying on NADH in order to pass those electrons. But so if you're making NADH through like the citric acid cycle and stuff, which takes place inside of the mitochondria, the mitochondrial matrix, then you have the NADH where you need it. But some processes are generating this NADH out in the cytoplasm. So just like the general interior of the cells, things like glycolysis are making your NADH out here. And in order to get that NADH into the mitochondria, well, you can't get it there directly. And so the malate-aspartate shuttle is one way that some of your tissues use in order to take the electrons that you generate from NADH and actually kind of sneak them into the mitochondria. And it does this using a series of antiporters and things like this. And so today I want to talk about this malate-aspartate shuttle as well as aspartate, the amino acid of the day. Your body used to be able to extract energy from a variety of different foods, from carbohydrates, so like sugars and starches, to lipids, so things like fats. And it needs to be able to convert these all into some sort of universal energy currency. And so, kind of think of an arcade token where you put a bunch of different currencies in and you get the same arcade tokens out. And in the case of our cells and energy, the arcade tokens are going to be ATP, adenosine triphosphate. I talk about it a lot more in another post, but basically in ATP, you have this really high energy bond. Basically, these phosphate groups are negatively charged, and so holding these negatively charged groups together is going to be kind of like clamping a spring. So this is going to be high energy. If you were to break the bond, you release the spring and you release energy. Think about if you have like a ping pong ball at the end of the spring, you release the clamp spring, the ping pong ball would fly off. And in the case of our cells, we can use that instead of flinging ping pong balls, we can get reactions to happen. And so we can do this because we're converting energy from all sorts of different sources into molecules of ATP. In order to make this ATP, however, because you're starting with different molecules, you're going to be going through different processes. And often, instead of directly making ATP, what these processes are doing is generating what we call like reducing equivalents, or basically they're generating things like NADH and FADH2, which are then going to be able to connect processes that happen out in the cytoplasm, so the general interior of the cell, to what's going on inside of the mitochondria. And specifically what they're doing is they're generating sources, those transporters of electrons that can be taken to the intermitochondrial membrane, to this place where the electron transport chain is going to be located. And so this is called like oxidative phosphorylation. And what it's going to do is it's actually going to be able to generate ATP. And this is where the big payoff from ATP making actually occurs. So in order to get, so you basically in processes like glycolysis, so when you're initially breaking down sugars, that's going to happen in the general outside inner part of your cell called the cytoplasm. But then the main energy making is going to take place in the mitochondria. So the plural is mitochondria. These mitochondria are basically these little cellular compartments inside the cell. And there's actually little compartments inside of those compartments. So mitochondria actually come from like a really, 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 really long time ago, like a precursor of our cells, swallowed a bacterial cell. And as in doing so, you end up with these compartments where you have these two membranes. You have this inner membrane and this outer membrane, and then you have this inner membrane space. And so the inner compartment of the mitochondria, this is called the mitochondrial matrix. And this is things where like the citric acid cycle happens here. And I talk much more about the citric acid cycle in other posts. It's sometimes called the Krebs cycle or the carboxylic acid cycle. And basically, this is where you are going to be generating the bulk of your NADH as well as some NADH too. And what's really great is it's happening in the mitochondrial matrix, which is the part where the electron transport chain is actually where the NADH needs to get. So inside the mitochondrial matrix, this is where the NADH is going to kind of pass the electrons that it's taken to the electron transport chain, which is the complexes which are embedded in this inner mitochondrial membrane. These are going to pump it out into the inner membrane space. So the inner membrane space is going to be the space in between the inner mitochondrial membrane and the outer mitochondrial membrane. And remember, all of this is happening inside of the mitochondria, which is happening inside of the cell. And it's outside of the mitochondria in the cytoplasm where the glycolysis is actually going to happen. So where that initial NADH making from the sugar breakdown is going to happen, as well as some other NADH from other processes. And so the bulk of the NADH, remember, is being made already in the matrix, which is where it needs to be. But if you have things happening out here where you're making NADH, well, now you're going to actually need to get that NADH into the mitochondrial matrix. And this is what you can't do directly. And this is why we're going to see that we're going to need this transporter. But first, let me back up and talk a little bit about the NADH. So basically, NADH, this is going to act as an electron carrier, and it's going to carry electrons in its reduced form to the electron transport chain. So you'll see NAD plus is the oxidized form and NADH is the reduced form. More on redox and other posts, but basically you can remember the mnemonic. Oxidation is loss of electrons or electron density, and reduction is gain of electrons or electron density. So in NADH, in the case of NADH, this is going to be the reduced form. It's gained electrons. In this NAD plus, this is the oxidized form. It gains electrons to become the reduced form. And much more on another post, remember, but this often when we talk about redox and biochemistry, a lot of times if something is reduced, it'll have you'll see an H on it. And if it's in the oxidized form, you won't. And so much more on this other post, but basically an H typically comes, you can get it when you form a bond to an H, you kind of can steal its electrons more easily. And so it's like you're gaining electrons. And so much more on this in other posts. I'm not going to go into it here, but I do want to point out that in the case of NADH, you can see that you're going from like a plus to a non-plus. And this is showing you like you're gaining an electron. You can remember there that you're gaining an electron. Electrons are negatively charged. This is going to balance out the positive charge. The big change between the NAD plus and the NADH, the change that matters is going to be that electron. You can see that you're also going to be gaining a proton. You're gaining a proton and two electrons. What this actually is this thing called a hydride. So we've seen proton transfers before where we're just transferring a proton. So just the hydrogen nucleus with just a proton. And normally we can also transfer in hydrogen where we have a hydrogen and an electron. A hydride is where you have a hydrogen and two electrons. And this is pretty rare, but it happens with this NAD plus and this NADH. So you're going from NAD plus to NADH. What you're doing is you're transferring a proton, yes, but that proton is going to be balanced out by having two electrons. And so overall, you're getting a net gain of an electron. And this electron can then be used in the electron transport chain where it's going to be passed from thing to thing. So you can see that in this complex one, the NADH is going to be converted back into NAD plus. You're taking that electron from NADH and then shuttling it through these complexes where it can get passed off from thing to thing to thing. You'll also sometimes see FADH2 and FAD, which is a similar case. You might also see NADP plus and NADPH. This is going to be a similar reducing agent, but it's regulated separately from NAD and NADH, so you can kind of keep different pools of these because, as you might imagine, you can regulate the proportion of NAD plus to NADH, and similarly NADPH and NADP plus, in order to kind of regulate which direction reactions are going to go. Are you breaking down energy, taking those electrons off, or are you building things up, putting electrons back on? And so NADPH is made through the pentose phosphate pathway, and it's mainly involved in the building of molecules, so anabolism, as well as like redox regulation, whereas NAD plus and NADH, this is mostly involved in catabolism, so breaking down molecules. And it's that breaking down molecules that we're talking about today, and using the energy from breaking down molecules in order to produce ATP. And so in order to get to this NADH, we break down foods through processes like glycolysis. So glycolysis is the initial breakdown of glucose, and this happens out in the cytoplasm. And from glycolysis, what you end up getting is pyruvate, which can then get converted through oxidative decarboxylation and to acetyl-CoA, which can then enter the citric acid cycle. And then when you enter that citric acid cycle, well, here you're going to get the big payoff of the NADH. From glycolysis, in addition to getting that pyruvate, you're also getting ATP to ATP and to NADH. And so the ATP, well, you can just use it as it is out there, but in order to actually make any ATP from this NADH, you're going to need to get it into the mitochondria. And you really, really want to get it into the mitochondria because that's where the big payoff from energy making is going to take place. So you might look at this and you might see, oh, cool, ATP, but just NADH, right? Well, for each NADH that goes in, you can actually get about 2.5 ATP. Because for each NADH that goes in, you're going to pump about 10 protons out of that inner membrane compartment out of the matrix into the inner membrane space. And it takes 4 protons coming in for 1 ATP to be made. So you have 10 protons, you take an NAD, you pump out 10 protons into here. And now remember, those protons are going to build up in here and they're going to flow through this pump. And when they flow through this pump, it's going to be driving it kind of like a dam, causing the conversion of ADP and inorganic phosphate, so just like a free-floating phosphate, into ATP. And so you're going to be taking NADH, passing its electrons, using those pass-offs to pump protons out. You get a pumping out of about 10 protons per NADH, and then for about 4 protons, you get an ATP. So you end up getting about 2.5 ATP per NADH. So although with glycolysis, it might not seem that exciting that you're getting NADH, like you're more focused on, oh, we get ATP. Well, you get the NADH that you can then convert to ATP. But in order to do that, you have to actually go through the oxidative phosphorylation. You have to go through that electron transport chain, not to mention all of the, you have to go through the electron transport chain. And so we need to be able to get that NADH in there if we want to get benefit out of it. And so this is where that malate-aspartate shuttle is going to come in, because it turns out that NADH can't get into the mitochondria. And so we need to use a workaround. So this mitochondria is going to be very, very picky about what it lets in and out. You might imagine, okay, well, it's got to be really have tight control over those protons, right?
because it needs to be able to keep this buildup of protons and keep it so that you have lots of exits out of the inner mitochondrial, out of the mitochondrial matrix, but you only have this single entrance. So you don't want these protons to just be flowing back and forth through this membrane, or else that's what we call uncoupling, where you don't have the protons basically being controllably transported, so that you only have the single way through. So there's actually things called like uncoupling proteins and like toxins and things that can uncouple this, so that the protons can kind of freely flow through, and therefore you're not able to make energy. And so there's actually, like, in brown fat and stuff, there's like uncoupling protons that kind of use it so you can like dissipate the energy as heat rather than actually making ATP. But in the case of most of your cells, this is what you want, is actually to make ATP. And so you're going to want this membrane to be very, very picky about what it lets in and out in the case of protons, but also in the case of other things. So this outer mitochondrial membrane, it's going to be less selective, so it's going to let more things through. But this inner mitochondrial membrane, in order to actually get into this mitochondrial matrix, is going to be very, very, very picky. And so things can't just flow through. NADH can't just flow through. Instead, things need to have dedicated transporters. So this could be like a channel, or this can be a pump of things like this. They have to have a dedicated path where there's some sort of protein that's making a channel or a pump to allow them to get through. And this way, your mitochondria are able to keep very, very tight control over what goes in and what goes out. In addition to protons, is a lot of other molecules, in part because the mitochondria, because it's doing all this metabolizing, this making and breaking of molecules, kind of like what we talked about in the case of NAD plus and NADH, where you can kind of regulate the proportions of the two in order to drive reversible reactions in different directions, or in order to have like one product inhibit the making of another product. In order to do all that, you need to keep tight control over the various products. So you need to have this membrane be very, very picky. And so your cells aren't going to just let NADH through. And so instead, what they're going to do is they're going to use a workaround. And this workaround is going to be clever, and it's going to pass the equivalence of those electrons from NADH into the mitochondrial matrix, where those electrons can then be converted back into NADH, which can then be used in the electron transport chain. All of this without actually taking the NADH into the matrix, but instead relying on separate NAD plus and NADH pools inside and outside of that inner mitochondrial membrane. So they're relying on different pools of NAD plus and just transferring the electrons from one thing to another. And the way that they're transporting this electron is in the form of malate, which can be converted between oxaloacetate and malate. And then inside, it's going to be converted back into oxaloacetate. Then ultimately, where this comes in is we're going to get aspartate. And so that aspartate can be pumped out. And so aspartate is going to be the topic of the amino acid of today. And before I get too much into how it's used here, I just want to mention a word about the naming. So you might see it named aspartic acid or aspartate. And so in aspartic acid, you basically have a carboxylic acid group where you have a carbon double bonded to an oxygen, double bond to an OH. In the carboxylate group, basically that's the deprotonated form of carboxylic acid. So it's given up a proton, and now it's in a carboxylate state where it's negatively charged. So this is one of the acidic amino acids, meaning that in its neutral form, it can act as an acid and give up a proton. In this case, so it has a carboxylic acid. If it deprotonates, it gives up a proton. It becomes its carboxylate form. And so we can call this aspartic acid and this aspartate. But in reality, people typically use the terms interchangeably. But in our bodies, we're almost all going to be in the carboxylate form, because it has a really low pKa. More on this in my other post, but basically the pKa is the pH at which half of it is going to be protonated and half of it's going to be deprotonated. If you go to a pH above the pKa, then you're going to have fewer protons available than it like wants in order for half of it to be protonated, basically. The higher you go in the pH, the fewer protons are around. And so if there's less protons around, you're going to be less likely to find it with being protonated. So you're going to be typically finding this in the deprotonated state, in this carboxylate state. And this is because you're going to have like resonance stabilization, which is going to make this really happy. And I talk about this a lot more in other posts. But basically, bottom line, aspartic acid and aspartate, you see these terms used interchangeably. Typically, you're going to actually be in this aspartate form, and this is how I'm going to be referring to it and how it's typically referred to when we're talking about the malate-aspartate shuttle. So let's get into a little more detail about this shuttle. Okay, so what we're going to be doing is we're going to have two antiporters. So an antiporter is a transporter that's going to let one thing through in exchange for another thing. And these transporters are going to be taking place or be located in this inner mitochondrial membrane. That's also the membrane where the electron transport chain is going to be taking place. And this is the more picky membrane, remember? So if we can get it through this membrane, we can more easily get it out between the cytoplasm and the inner membrane space. And it's really getting into the inner matrix that's going to be the hard part. And this is where our antiporters are going to come in. So there's a lot of different errors and things going on, but really what we're going to do is we're just going to be what we care most about is going to be this NADH, and all the rest of this is just to regenerate it. But we have NADH that we generated from something like glycolysis or something. Okay, so let's start here. This is a molecule called oxaloacetate. You see it a lot in biochemistry. You see it made in various processes, including the tricarboxylic acid cycle. So including that Krebs cycle, that's TCA, that citric acid cycle, whatever you want to call it. This is, remember, the place where we're going to be getting the bulk of our NADH. And this is actually going to be happening inside of the mitochondrial matrix, as well as being generated in other places. But you end up with, so you have oxaloacetate out in your cytoplasm that you can then convert to malate by reducing it. So remember, reduction is the gain of electrons. That's our rig and oil rig. And here what we're doing is oxaloacetate is becoming reduced. So this carbonyl group, this C double bond O, now it's becoming a hydroxyl group, an OH. And remember how I said that kind of a bond to hydrogen is often associated, but not always with reduction, or a loss of a bond to oxygen. So here you're losing a bond to oxygen, you're gaining a bond to hydrogen. This is because oxaloacetate has become reduced to form malate. And those electrons from that reduction have to go somewhere, and they're going somewhere. They're basically, that electron had to come from somewhere. That electron was coming from the NADH. And so now this is storing it. Now this malate holds the electron that we had put into NAD plus to make NADH. So now we've made NAD plus out here, and our electron is now in malate. Now this matters because unlike NADH, malate can get through, but malate's going to get through in exchange for an alpha ketoglutarate, which is going to come in handy later. And so this malate is now going to get transferred into the mitochondrial matrix in exchange for that alpha ketoglutarate. Once in here, now that malate can get converted back into oxaloacetate. So now we're going to reverse the process we just did out here. So this enzyme malate dehydrogenase, the cytosolic form and the mitochondrial form. So you have it in here and you have it out there. You have it in here and out there. And what it's going to do is an enzyme can catalyze or speed up reactions in both directions. So in this case, it's making it happen so that you have it be generating malate. And now in here, you're going to be generating oxaloacetate. And so when you have reversible reactions like this and things, this is where it makes a really big deal what concentrations are of the various products, and like the reactants versus the products, and which direction the reaction is going to flow. So out here, you're going to have it go from oxaloacetate to malate, and then in here, you're going to have it go from malate, get oxidized back into oxaloacetate. So oxidation is loss. This is losing electrons, and it's giving those electrons that it loses to NAD plus to make NADH. And now these electrons can go through the electron transport chain to make ATP. So these can pass those electrons off in the complex 1, which can then pass those off further, further, and further, pass off these electrons and pump out protons, which can flow back in to make ATP. So this is where the NADH, now you've taken this NADH that was out here, and you kind of made the equivalent of it in here without actually passing NADH through the membrane. So in it, we've basically taken this oxaloacetate, converted it to malate, and transferred it in. If we want to do this again, well, we need to regenerate this oxaloacetate. What happened in here was that when we took that malate in, what we did was we transported alpha-ketoglutarate out. And it turns out that alpha-ketoglutarate, we can actually use that to make oxaloacetate. But we're going to need to combine it with the amino group from aspartate. And so aspartate, we're going to take the amino group from aspartate and pass it on to alpha-ketoglutarate using an aspartate amino transferase. So amino transferase, basically you're taking the amino group from one thing and transferring it onto another. And often this is going to happen through alpha-ketoglutarate, which is basically going to be the glutamate without the amino group. And so if you give it an amino group, you're going to make glutamate. And you can give it an amino group from aspartate. And the reason why we're using aspartate here is because we'll see that we can actually get that through another transporter from the mitochondria. So we take this amino group from aspartate, transfer it onto the alpha-ketoglutarate. And remember that alpha-ketoglutarate was what we had taken out in exchange for bringing malate in. So now we can take this alpha-ketoglutarate and we can take this aspartate, we can make glutamate, which we don't really care about in this case, but it can go and do other things. Or what we can do is we can actually use it. We can then, the other product that is made is oxaloacetate. So basically that's the leftovers. When you remove the amino group from aspartate, you get this oxaloacetate. And now this oxaloacetate can be used in order to make and get reduced again in order to make malate, which can then go in, and then you can regenerate this. But we're not quite done, because once we go through here, we still have this oxaloacetate that we've made inside of the mitochondrial matrix. And we still need to be able to get this aspartate out here so that we can, if we really want to keep this cycle going. So it's not that you don't have like aspartate out here already, but in order to keep this cycling, you have to be able to regenerate everything that you've cycled through, except for this NADH and NAD+. So this is the only thing that we want actually changing. We want to keep the concentrations of all of this other stuff. In order to keep this cycling, just cycling, you would need the concentrations of all this other stuff to be the same or to be like constant. And so you're going to be able to replenish everything. And so in order to replenish this aspartate, we need to be able to get it from inside. We need to be able to make it inside the mitochondria and then pump it out of the mitochondria in exchange for that glutamate. So now we see the opposite of this going on. So we have the same, so we have aspartate amino transferase again, but in this case, what we're doing is instead of transferring amino group from aspartate to alpha-ketoglutarate, we're actually transporting the amino group from glutamate onto oxaloacetate in order to make aspartate. And then the aspartate can get transported out of the mitochondria. And in order to do so, what's going to happen is that this is going to get transported in exchange for getting more glutamate in. And in this way, you're going to be regenerating these various processes. And this is also where you're getting the alpha-ketoglutarate that you're going to transport out in exchange for that malate. So you can see that this is all interconnected, and this is all going to be done to recycle these various products, when really all that's happening, all of this is trying to do is take an electron from NADH and get it inside the mitochondria. But because this membrane is going to be so picky, you're going to have to be going through this like convoluted method where you're transporting different things in and different things out. So in sum, what we have is this NADH equivalent being regenerated. And how this is happening, let's go through all these complicated arrows again. So we'll start with the NADH, and we're going to be taking the electron from NADH, giving it to oxaloacetate. Oxaloacetate, this is what this is doing, is it's going to be reducing oxaloacetate. So reduction is gain, you're gaining an electron. You're going to be taking this oxaloacetate. If you look at this carbon, you can see you have a carbonyl, and here you have a hydroxyl group. You've lost a bond to oxygen, gained a bond to hydrogen. This is going to mean this is showing you this is the reduced form. When something is reduced, it's gained electrons. And so now what we're going to do is we're going to take that electron in the form of this reduced molecule and take it into the mitochondria. Once in the mitochondria, that electron can then get passed back off to NAD plus to regenerate NADH. But now this is with a separate pool of NAD plus and NADH that's located inside of the mitochondrial matrix as opposed to outside in your cytoplasm. And so when you do this, now you're regenerating oxaloacetate. Now, in order to get that malate in, you had to pump something out, and you had to take something out. And what you took out was alpha-ketoglutarate. So this is the malate alpha-ketoglutarate antiporter. So an antiporter, you're taking one thing in exchange for taking another thing out. We're taking malate in and taking alpha-ketoglutarate out. And we'll see that this alpha-ketoglutarate is going to be used in order to regenerate the oxaloacetate out here so that you can keep this going. The oxaloacetate that you generated in here, this is going to be used in order to make aspartate. And aspartate is what you're going to need in order to get glutamate from out here into here. And so this oxaloacetate is going to be combined with, so oxaloacetate is going to react with glutamate and aspartate amino transferase in order to make the aspartate that you're going to pump out, which you're then going to combine with alpha-ketoglutarate in order to make the oxaloacetate to get the cycle going again. So this oxaloacetate that you make in here, in order to actually convert it into aspartate, you're going to need to use glutamate. And this glutamate is going to be transported through that second antiporter in exchange for that aspartate. And so in this second thing, you're going to have this oxaloacetate react with this glutamate. This is going to give you alpha-ketoglutarate and aspartate. And the alpha-ketoglutarate, this is going to go through, this is what you were exchanging in exchange for the malate in this first antiporter. And then this aspartate, this is what you're going to be exchanging in the second antiporter. So you're going to be, through this antiporter, you're letting malate in and you're letting alpha-ketoglutarate out. Through this antiporter, you're letting glutamate in and you're letting aspartate out. All of this is just to regenerate these carrier molecules. So if you follow all these arrows, you'll see that you're going to be regenerating everything. The only thing that you're changing, the only thing you need to keep supplying is going to be the electron. So you're going to be needing to keep having this NAD, this be regenerated, because here you're going from NADH to NAD plus. And now in order to regenerate this NAD plus, well, you're not going to find that on this pathway. Instead, you're going to be finding that NADH. You're going to be finding that coming from various processes such as glycolysis. And so this is how you can regenerate this NADH. And then remember that most of the NADH that you're making, that you're going to be using for this respiration, for this electron transport chain, is actually going to be made in the mitochondrial matrix. And so we don't even need to worry about this for most things. So not all the cells in your body actually rely on this process. The liver, heart, and kidneys, this is going to be, they're going to use this as a main form. So it's really, really important. Some tissues use alternative like shunts and things like this. Aspartate can also be used in other metabolic processes, including this like Krebs bicycle. Basically, the citric acid cycle is really, really useful for doing things like break down amino acids. But you can generate some nitrogenous waste because amino acids are going to have that nitrogen group. And this nitrogen group from different amino acids can get transported onto oxaloacetate. And this oxaloacetate can then be used through this urea cycle to get rid of that waste safely. And aspartate is going to basically, when you pass an amino group onto oxaloacetate, you get aspartate. And so that aspartate is going to play a key role in order to basically that can get shuttled out of the mitochondria and get converted into arginosuccinate, which can then enter that urea cycle to get removed that way safely as urea, as opposed to like dangerous ammonia and stuff. So that's called the aspartate argininosuccinate shunt. And yeah, so it relies on being able to make aspartate that gets shuttled out of the mitochondria. So that's aspartate. Sometimes you might see it written as aspartic acid. Typically, people use these terms interchangeably because this side chain can interchange between the carboxylic acid and the carboxylate forms. All that's different is a proton. So this is just going to be pH dependent. And at the pH of our bodies, we're going to be way above the pKa. Bottom line, it's going to typically be in this deprotonated carboxylate state, which we call aspartate, but you might see people just use the terms interchangeably, but technically that is the difference. Aspartate is going to be very, very useful for things, including the malate-aspartate shuttle, which is going to allow you to get NAD or the equivalent of NADH into the mitochondria, connecting energy making processes like glycolysis that happen in the cytoplasm to the inner mitochondria, where the matrix, where you're actually going to get the energy making happening through oxidative phosphorylation through the electron transport chain.